We will now begin with our another lecture delivered by Dr. Imadul Islam. In this lecture, Sir will walk us through how drug discovery begins, what initiates the process, and who the key contributors are in this complex journey. May I now please call Dr. Imadul Islam on the stage.
Just thinking this will go. Assalamu alaikum everybody and good afternoon. Sorry for being late. We were meeting with your Vice Chancellor, so we cannot leave her. I will request everybody, those who are sitting on the back, please come forward so that you can come closer. That's better. Then you can hear me properly also. I was never backbencher in my life, so I want everybody to be front bencher. Yes, that's the reason that if you are closer to screen, it is better. No, I think that's okay. But they can see right
Yes,
at the
sir,
screen, and I can see this also. So that's okay. That's not a problem. Okay, so the topic of the talk today is art and science of drug discovery, and I will try to set up the stage for next 4 days, 5 days you have. In this lecture, I'm going to talk about almost all discipline. Dr. Azhar Aziz will also talk about some aspect, and Dr. Zainuddin will also talk about some aspect. I'm not going to go into detail. I will just give you a holistic view of the drug discovery in this lecture, and then I have 5 more lectures, and I cover some of the part during that. So let me first introduce myself. My name is Imadul Islam. I work as a Deputy Director of Drug Discovery and Director of Pharmaceutical Analysis in King Abdullah International Medical University Center, Medical Research Center. And before I was in US almost for 25 years or so. I worked in different companies, Bayer, GlaxoSmithKline, Bristol Myers, Lundbeck. And I was, by training, I'm a chemist. I have a PhD from IIT Kanpur and Allahabad University in chemistry. So that's my background, and I have a long experience in drug discovery. A couple of products, they are either going on market or they are in phase clinical trials, 1, 2, 3. So that's how my experience goes along. So the presentation outline will be a little bit different than what usually scientists do. A prologue, monologue, introduction, process of drug discovery, lead optimization, then preclinical, phase 1 and 4, and then epilogue, it means last part. This is all we are going to talk in next 4 days, inshallah. So that's the topic. I will just skip that. Uh, so I want to give a idea of what we do actually in KMARC. So we do, it is an integrated drug discovery center in Saudi Arabia, and we do target identification. I'm going to talk about that also. Lead identification, lead optimization, then in vivo, ex vivo profiling, Dr. Zain and Dr. Aziz will talk about little bit, and I will give a little background of development part also. In Saudi Arabia, we don't do any development, so that's why I grade it out. I was just appointed again by the Minister of Health Affairs, reappointed me as a director of PSC, so this is a 2-year term again. I was there for 2 years and 2-year term again. So monologue is, what is drug discovery? I have 40 minutes, right? 40 minutes. Okay, so I just want to see the time so that I don't go over time, inshallah. So monologue is knowledge. What is drug discovery? It is knowledge, experience, and creativity. So this is a page, okay, and there are some dots you can see there. So this is knowledge, okay. Dots are spread on a blank page, but if you have experience, you can connect the dot, and it looks like a picture, okay. And if you have creativity, same dots can be connected into a work. You can see a artwork. Drug discovery is not different. It is the same. If I go back, and this is the knowledge, you see all kind of elements are there. Chloro, fluoro, bromo, iodine. Everyone knows that, the periodic table, right? If you have a little bit background of chemistry, you know that. Then if you have experience, okay, you can connect hydrogen to carbon, it becomes methane. You can connect sodium to carbon, it becomes sodium chloride, which we use every day, eat every day. And if you are creative, then you can connect the proper dot to proper place. तो इस तरह से जो है, you can develop a drug called Januvia, Lyrica, or Leda. So this is the drug discovery in nutshell. The whole parameter will be going around that in next 4 days, okay? So you have to be knowledge, you have to have little knowledge, experience, and creativity. So this is drug discovery. And actually, last part is not showing. So drug discovery is science, art, and it requires knowledge and experience and creativity. So that's the last part which I have written there. Okay, and if you are more creative, then you can see, actually it is not very clear somehow, but if you can little bit visualize that. So the same molecules, they can be like different shapes. With carbon, hydrogen, nitrogen, it can be एक बकरी की तरह से दिखाई दे रही है, एक आदमी की तरह से दिखाई दे रहा है. So this is another, part of drug discovery, that you have to connect these dots. And when you connect the dots, you get a product. So how beautifully Jean-Marie Lehan, he said about drug discovery and the chemistry, that building a legacy with knowledge, experience, and creativity. Atoms are letters, molecules are words, and supramolecular entities like protein. Mr. Sayyid Ahmad, he talked about vaccines, right? And that is the chapters and sentences. So he was the one who gave this statement, and it's so true these days in drug discovery. Okay, and then the discovery in drug is architect don't discover books. Okay, authors don't discover books. Architect don't discover buildings, and scientists don't discover drugs. So what they do and what we do, we invent, design, engineer molecule to preserve biological system. What is biological system? It is proteins which you are generating in your body. It is creative, collaborative, informed process built on all kind of technical skills and excellence. All of them you are going to listen, inshallah, in next few days. Everyone is here who has done some work. New medicines are not just lying around. And last one is drug are not magical or propelled. Their behavior should be explained by rational PKPD. So Dr. Zain, who was sitting here, he is a PKPD scientist, and he's going to explain a lot about pharmacokinetic and pharmacodynamic. I wish that we have this screen proper, maybe inshallah. But I'll keep on reading that. So if you do this work, and if you discover a drug, better than, okay, yeah, what I see is okay, I'll focus on my screen, that's better. And if you can see there, it is good. So I cannot see here all of that. So top 10 small molecule pharmaceutical by sale, this is 224 data. And you can see that how big numbers are. The Eliquis, there's a one small molecule drug, it is sold 20 billion per year. And somebody has made it, actually it was made by one of my colleagues in Bristol, Massachusetts, and she was rewarded for that. And not only that, this drug is on market, it saves lives. So a lot of people are saved. This is an antithrombotic and blood pressure drug. Similarly, Bactari, Jardine, Tirifata, Farxiga, Artrestos, Anti-D, and all those things. You see in terms of billions how big they are in sales. So what drug discovery scientists, they discover these drugs. And they do by chemistry, by pharmacokinetic, and all other parameters, they use that. So this is just, I just showed it there. Numbers are maybe changed, but you can see that these drugs are sold in a very high volume and with a lot of money around. And similarly, you can see other drugs also, so I'll just skip. And then drugs are not only small molecule, there are biologics also. Biologics means protein, vaccines. So like Keytruda, a cancer drug which is sold by Merck is $29 billion. This is like a small country budget, 29 billion, one drug only, one drug. If you discover something in AMU, AMU will be on the world map. So just aim for that, inshallah. That's the thing. And this is not very difficult. Dupixent, Drazila, and Opdivo. Actually, Opdivo, I was partly involved in developing that drug in Bristol, Myers Squibb. It was launched when I was there. These are the give back to you, give back to your community that these drugs are available in the market. So with this introduction of drug discovery, I will go to what we are going to talk in next 4 days. The drug discovery process is a target identification, hit identification, lead optimization, and late lead optimization and development. And if you do that, you will earn a lot of money in terms of dollar, and since I'm in Saudi Arabia, I'll say SAR also. So these are the topics we are going to cover, most all three of us, inshallah. And I will talk about target identification, hit, lead, all those things, and what are the aspects of why we do that, and what are the roles for scientists in that, in drug discovery. So drug discovery process timeline is quite long. If I go to next, it is like 6 to 12 years. Yes, I know people are going to ask that AI will help. I will talk about in other topic, that how AI can help. I will not talk today, but this is the process you have to remember, and we are going to talk a lot about this process. So from idea to medicine, it is 6 to 12 years. And the life of the drug is very short because the patent is only for 17 years and sometimes 20 years. So, you know, you spend billions of dollars and then you get only very short time. But think back on my slide where 1 year it was sold for 13 billion. So why not, we should not discover the drug, okay? And this is normal, typical. I worked in a project which was like within 5 years we had a drug already. So this is, I just summed up everything, okay? ये average कर दिया है हमने कि इस तरह से होता है ये. From idea to medicine, 6 to 12 years. So it is a very tedious process. We don't have to get scared because otherwise we will not have any drug in the market, what we are taking. So company makes a lot of money out of it. And academic institution like AMU is maybe part of that. So when we do start with that, we look at the patient, why he is sick. Then we try to find out the target, what was the problem with him. And then target identification takes place, gene, protein, Dr. Azhar will talk about a lot. Then I come in picture, active compound, then we optimize the compound, then we go to preclinical, and Dr. Zain will talk hopefully about some PK/PD. In clinical trial, phase 1 and 2, we will touch about that. I will touch in one of the talk about that how it works and what is the requirement for that, and then registration and market. So drug discovery, so attrition, now, you know, it is not easy, as I told you. So how many compounds usually we need to get a drug? 10,000 molecules. Not always, okay? 10,000 molecules, not always. So hit, we get like 10,000. Then we zone him to 250 molecules. Then next we go to 10 molecules after attrition and after doing studies, one after the other, hit identification, lead optimization. In preclinical, we put 10 molecules. And then in clinical phase 1 to 4, sometimes 4 molecules, sometimes 5 molecules. And if you're lucky, you can put only 1 molecule, which will be really good. And then go to registration and we get 1 molecule, which is in the market, okay? So it has a very high attrition rate, and the cost of developing drug is 0.3 to 1.5 billion. Some people say it is more. I'm not going to debate on that. It depends which area and how lucky you are. In my experience, because I worked on many projects, uh, from 500 to 3,000 molecule we made, and we discovered a molecule which was good for some disease. Uh, when I give a case study at that time, I'm going to talk about a real molecule which is already into phase 2 clinical trial. Okay, so key role of chemist. I told you this morning also I am a chemist, so I will talk more about chemist than other sections. So chemist has a key role in the drug discovery. So you can see in this slide, so there are medicinal chemists who are synthetic chemists, and process chemists are also chemists. I'm not confusing you, but this is like we learn. There is no degree for that. I'm an organic chemist, but I'm a medicinal chemist because I learned at the job, medicinal chemistry. Similarly, a process chemist is an organic chemist, but he learned job at the how to make tons of molecule, tons of kilogram. Okay, think about it. Aspirin is made at least like 179 metric ton every year. So where it is made. So this is the process chemistry job. Then somebody was asking this morning about computational chemistry. Yes, we use computational chemistry also. And analytical chemistry, Dr. Zain will talk some about that. So we use a lot of analytical chemistry to identify our compounds, whether they are right or wrong. And those things are done by all these. So this is the role of chemist in the whole drug discovery process. So they have distinct roles in the drug discovery process, but significant overlap with each other. So I can be a process chemist and I can be an analytical chemist also at the same time. So that works for me. And close communication is required between all disciplines. So here in lot of departments, industrial chemistry, organic chemistry, and analytical chemistry, they all have to come together to have a company here. So that's very important. So the role of medicinal chemist. Since I'm a medicinal chemist, so I will just give a little bit background. I'm not going to talk about any chemistry, just give you what we do and why you should make a career in medicinal chemistry also. That's another part. So what we do is we design a molecule, novel compound, design synthesis of this molecule, perform synthetic sequence to produce target molecule. So designing novel synthetic route and novel chemistry. We purify the compound, analyze it by spectroscopy data. We register for submitting compound for relevant assays to biochemist. Then designing a better compound, because it is not necessary that you get the first compound which will be really active. So you have to optimize it. Contribute to patent protection and protect the drug molecule. Dr. Rizvi is here. We talked about that thing. It's very important. Don't publish. Dr. Syed Ahmed also said that, and I am also saying that don't publish until you patent it if you see there is a potential, because you may be losing $1 billion. And we lost some, in some cases we lost. So talk to people who are knowledgeable in IP arena, and they can tell you whether it is good or bad to publish. Don't publish before you get into that. Any idea, this is not about the molecule only, any device, any sensors, please get in touch with Dr. Rizvi, and he will connect you with the right people. Even you don't have any idea, even you don't have any device, but you have some idea. Ideas are also patented these days. So he will tell you more about that how idea can be patented, and you can patent the idea also without doing anything. But you should have the novel idea. Okay, preliminary route for optimization in conjunction with chemical development. So that's the role of medicinal chemistry. Additional to that, we have some more rule which is called PET reagent or positron emission tomography reagent, which are used for the treatment. Imaging agent, like if somebody has some kind of disease where imaging is required, so we develop, as a chemist, we develop imaging agent also. We develop a specific tool compounds for drug discovery also. If you need a compound which can be used for some purpose, we can design and we can at least suggest and diagnostic reagent, which are, diagnostic labs are there. Most of the molecules or compounds or tests which are used there may be developed by the chemist. So that's the purpose of role of medicinal chemist. Okay, so I'll talk one by one, every analytical chemist, what they do. So they support the molecule in drug profile. So when you have drug, if you think you have a drug, so you have to go to an analytical chemist and see whether they are the real compound or something else. So this is the part of their job. And so they develop analytical and spectroscopic method. They demonstrate robustness of method also. And this is all required for filing a regulatory office, like in India it is, I think, a drug authority. In Saudi Arabia it is Saudi FDA. In US it is FDA. So we have to file, every simple thing they need to know whether what you have done, because they need to know that it is the real compound. Just if you type, just last few days, if you type Professor Kim Janda's name in Google, they proposed a compound 20 years ago, and suddenly they came to know, and he's a professor in UC San Diego. So he discovered that the compound which was marketed for 20 years for at least $120 billion has a wrong structure. Can you believe that a wrong structure was already sold, and he proposed a new structure, and that was right. So it is a big news, actually. Professor Kim Janda, his photo also came all over the media. So it is very important that you should know what you are making. Provide structure elucidation facilities and assure purity and safety of drug. That's very important. Please do not, take it, that it is pure. Just I know it is pure. No, it has to be done properly with the robustness of the procedures and the technique. So this is the role of analytical chemist. Computational chemist, there are several questions were coming this morning, and then afternoon somebody asked me, so I'll just put everything together. Yes, we use computer also. We use computer power to discover drug. We use computer power to optimize drug. So apply computer power to solve the problem, because they can visualize which you cannot see from open eyes. So computer can look into that and tell you that, okay, what you are looking at is right. And usually there are programs which are already known, Schrodinger, MOE. Recently you might have heard about AlphaFolds 1, 2, 3. These are all programs which are done by computational chemistry, and AlphaFold recently won 2 years ago, it won the Nobel Prize also. This is a computer program run by Google and other professor from UK. He is the youngest, I believe, 35-year-old, and he won a Nobel Prize by developing this algorithm. So we don't use that, but we use some other programs. We use molecular modeling technique and physical chemistry principle. We calculate energy conformation of drug molecules and calculate binding energy of the drug and proteins. So I work in protein and small molecule both. So these programs are meant to discover drug and to refine your finding, actually. It is not the program which will give you a drug, but it will refine your drug molecule. And you can work in 2D and 3D. There are experts there, and they generate new targets and non-obvious relationship, and they calculate physicochemical properties, which is these days getting a lot of tracking, because physical properties were not earlier used properly. Now they are looking into that also. So this is a computational chemist job in the drug discovery. And we have, you can look into the molecules in 3D and 2D, and how it rotates in the space, how it binds to a target molecule, which is usually protein or ion channels, or GPCR, which is called G protein coupled receptors. So these are all disease targets, actually. So you can visualize through computers. So this is the task of the computational chemist. So lead optimization. So molecular space is a very big space, okay? And you have to navigate into that, and I will show you what happens once we start doing a drug discovery. So this is the activity X, okay? Then this is a selectivity over Y target. And then acceptable ADME property. So ADME is absorption, disposition, metabolism, excretion. So you have to meet all those, and your molecule should be where? Here. So now how you reach there? So you can see how the space is small, but the task is very big. So you have a molecule which is active on X, it is selective on Y, and then it is acceptable ADME. Everything should be put together and it will go into that small blue place. So we start. So we started with, okay, we got a compound or which I got a protein or a peptide, which is a lead compound, okay? We'll talk about lead in my next lectures. What are the lead compounds? Then we try to optimize it and get to the blue part, but it just goes away and goes to target X. Okay, so now it is out of one part. Now we try again, make more compound, but it does not go to blue part. Then we try to make again, and it goes beyond, okay? Then we keep trying, keep trying, keep trying, so it comes into the acceptable, but it does not have the activity X and selectivity at Y. And finally, it is coming closer, and then it goes back again. So this happens all the time in drug discovery. We struggle quite a bit, but sometimes we are lucky, we don't have to struggle, and we get the molecule very quickly. And finally, we get the molecule which we want. So this is the molecule which is important, and that's how the lead optimization takes place. So only a small molecule, a small portion of chemical space has the desired properties or desired balance of properties. The medicinal chemistry's role is to find that molecule that is exist in that blue space. Okay, and then and it is supported by all kind of people. I showed you analytical, computational, ADME, molecular biology, pharmacokinetic, pharmacodynamic. Then we get a molecule there. So this is the lead optimization process. And chemical tractability enables us to perform effective lead optimization. So we track the molecule all over. Every time, wherever we go, we track the molecule that it is there. Okay. Now, I hope that you see there. So what I say here that it may not be possible to find a compound meet all the criteria which I told, okay? And it may not be possible for any compound to meet the criteria. It means you have to close the project. So that's very simple, okay? So you do work, and we have closed the many projects, but sometimes we use those projects for other purpose also, teaching purpose also. So that's the drug discovery or lead optimization. So as I told you that I'm going to give you a whole spectrum what we are going to do in next 5 days. So we reached up to lead optimization. Okay, so in the lead optimization, you need the molecule. You need a molecule in this bottle. This is a vial, okay? We need 10 milligram of molecule. We have to make that. Why we need 10 milligram only? Because Dr. Zainuddin want 1 milligram to see whether it is pure or not, and he will use a method called LCMSMS. And sometimes he might ask 5 milligram to dose in the animal. So we need that molecule. So chemist's job is to do a small scale synthesis for identification and characterization purpose. Okay, what happened? Okay, came back. Oops. I think I need some help here. Yeah, presentation mode. Oh, it is here somewhere. Okay, I can go from here. Okay. Okay, now it is better actually. It is showing full screen. Earlier it was not showing full screen here at least, but it was showing there. Okay, now it's better. So, so these 10 to 15, 10 milligram, and that is where this drug start. 10 milligram of compound you need for identification and characterization. Then using the analytical technique like NMR, IR, mass spectroscopy, HPLC, everything I think I'm sure that Aligarh Muslim University has that in the chemistry department or other departments. And use in vitro assay and enzyme cell-based assay. I think Dr. Azhar will talk more about that a little bit earlier. Uh, why I put my picture there? Can anybody tell? Why not? Because I still work in lab, even today. So that's why I put my picture there that I was working lab and somebody took a picture of that, a photo of that. Actually, there was an iconic photo I forgot to bring. Me and Professor Casey, we were talking in a conference room and he got idea. He said, come outside. And we had a coffee cup in our hand and he started writing a structure on that and somebody took a picture and sent it to Professor Bandar Ganavi. And this became an iconic picture in the Kmart that these guys, so I just told Dr. Bandar Ganavi that if you see any chemical graffiti, it means it is me and Dr. Casey. he was laughing. So this is what, you have to be in the lab and you have to work sometime harder. Sometime take it easy. We take it easy. We go for fun. But we wait our hand is still in our lab. So that's my message to you for budding drug discovery scientist. So laboratory scale reactor, this is a process development kilo scale reactor. So from 10 milligram, we go to maybe 100 gram to 1 kilogram. Why we go there? Why we directly go to tons? Because then we have to dose in animals. So that is called animal studies. We will talk in our couple of talks, what animals we use, where we use, why we use. We will discuss all those things in the next few talks. This is a reactor. I just want to show you the picture of that this gives you a kilo scale, and we make kilo scale. This may not be GLP material because it is going to animal, but if you are going to human, you have to make a GLP material that is required by FDA. So this is also a chemist's job to do that. Then you can see these are the plant, and Dr. Zainuddin can tell you whether it is from your company or I think it is. I took this picture from your company where you are working right now. Could be. So when we make a tons, multiple kilogram and ton scale, then we go to reactor which are already half buried in the ground, and you won't believe that what you take a small pill is made in about 200 metric tons sometime. 200 metric tons is a lot of drug, and chemists and chemical engineers are involved to make that. And sometimes process is too long and it takes very long to make also. So you appreciate that they are working for the humanity. So now I go to preclinical development. So this is our discovery part. Now you are in drug discovery. There is a function I showed on my first slide that there is a preclinical part also. So preclinical is the CMC, chemistry and manufacturing that requires formulation. You heard today with Mr. Ahmed about formulation of vaccine. That is part of the CMC. And what you use, what you don't use, all those are very, very critical once you make a compound. Whether it will be used by, it may not be amenable to a human being even. So that's very important. And a detailed physicochemical property characterization, impurity profiling, all those things you have to do that. And these are used for animal studies, and data will be used in human phase 1 also. Then benchmark in vivo model. So in vivo model is once you have a compound in vitro, in cells and enzyme, you have already tested, now you have to go to certain animals. So in vivo models are the diseased model sometime, and it is validated model. We use in animal models like mice, rat, rabbit, marmoset, and many times dogs and rabbits also. And then we go to monkeys also. Monkeys are very difficult to do because it is highly regulated, in US at least. In Saudi Arabia, we don't have any facility for monkey right now. but we have facility for up to rabbit and marmoset. So we do that. So ADME profiling, which is done by Dr. Zainuddin, optimize analytical method development. You need a specialized equipment for doing that, basic pharmacokinetic, pharmacodynamic, oral bioavailability, determining metabolism of drug, that's also important. There are many drugs, I will talk some of them, just give you the name that you go and look at and that. And mostly they are done in mice and rats, sometime in non-human primates, which is like monkeys. Then preliminary toxicology, this is also important that you need to take a drug which is safe. So we have to do toxicology and we have to do how much dose you need. You cannot take a drug for 1 gram. Maybe you need just only 10 milligrams. So we call it maximum tolerated dose. Repeat dose, non-GLP repeating dose, preliminary cardiovascular safety, pharmacology, all is being done in preclinical side. Several species are used both in vivo and vitro for these works. So this is also a huge amount of work which we usually do to get a drug to market. So human clinical trial, just I will give you a flash of that. So there are phases of clinical trials in humans. So okay, you got a drug, you went up to animal, everything is clean, no toxicity, it is efficacious, it is pure. Now we will start thinking about doing in human. So going from animal to human is another paradigm. तो उसमें कुछ और ही बात होती है, ह्यूमन में जो है वो कुछ और बात होती है. हमने सारा कुछ कर लिया एनिमल में, लेकिन सब कुछ ठीक ठाक दिखता है, बट समटाइम वी फेल देयर. So clinical trials are biomedical health related research studies in human. Clinical trial used to predefine protocol approved by regulatory authorities. In India it is CDRIR, Central Drug Authority, in Saudi FDA in Saudi Arabia, and FDA in the US. So there are 4 clinical trial phases. One is human pharmacology phase. Second, phase 2 is therapeutic exploratory phase. Third is therapeutic confirmatory phase, and fourth is post-marketing. Let's go by one. So phase 1 trial, first time in human. This is another, if my compound goes to first time in human, I'll be jumping on my feet, okay, that my compound went to first time in human. So we call it phase 1 clinical trial. In this case, we don't use a large number of subjects. They are all healthy subjects. They have to be paired, and actually these days I'm struggling to get human, healthy human volunteers in Saudi Arabia. We need Saudi healthy volunteers, and they are not coming forward. We have to do some trials. So this is a big thing for clinicians, that they have to do that. So 10 to 100 individual of both gender. We test safety of the molecule, a small molecule, and biologics both. Biologics means protein-based molecules. Determine safest dose, which is dose which has to be used, and we range the dose. The best way is administration, whether it is oral, IP, or any other way. Identify the side effect, which are not evident in animal studies. So that's why I said that humans, they showed up some of the side effects they showed up in human. The subject is monitored for several half-life of drug, and also determine the volume, distribution, clearance, and oral viability. All will be discussed in next couple of days. In phase 2 trial, done on most result of phase 1, most important phase is drug discovery. Here we use 20 to 300 individual or participant. We continue to monitor safety of the drug, it is safe or not. Begin the access of efficacy of certain disease. All participants receive same experimental dose so that we know it is working. And sometimes phase 2 is divided into phase 1 and phase 2, and that is the reason why we do that. Phase 2A designed to assess the dosing requirement, and phase 2B designed to assess efficacy of the molecule. Phase 3 trial is most important part, and 1 in 10 fails at that stage usually. 1 in 10 drug fail at that stage. So from beginning to here, you already spent about 200 million. Now you can fail, or you can make $1 billion. That's the reason for that. And that's why I put a little bit longer so that everyone can understand. Trial done on larger population from 100 to 3,000. The numbers depend on the therapeutic area also. In cancer, you don't get that many patients. But in cardiovascular these days and diabetes, everybody has at least one of them. Even 40, 30-year, 40-year-old, so you get a lot of patients. That's why the number is there. Can be multi-center trial, assessment of the effect of drug effectiveness, compare the new treatment, which is better than standard of care, side effect. So somebody already has a compound or drug already in market, but we still make because we can be better than them in many ways. Assess risk, outright the benefit, uses two more treatment group, experimental and control. Usually groups are randomized and sometimes blinded. These are the clinical trial part, and the clinician, he knows about that. So we have to do that to understand the activity of the drug on that disease model. Most expensive, time consuming, difficult part of the drug discovery. This takes most of the money. We spent 200 million up to here, and from 200 to 1.5 billion we give to our clinicians. And then new drug application is being submitted for the drug. Now regulatory authority comes in picture, and he is going to look your data from where I started, if you remember, target identification up to this place. Regulatory authority earlier, I remember when I was in Michigan and I saw a truck was going from my company, and I asked, what is that? He said, this is a data for all the molecule, all the studies we did. This is going to Saudi FDA. This is all data, because at that time we used to write by hand everything, and there was no CD and there was no flash drive. So it used to be like that. Now you can put in a flash drive everything and just send it to FDA, and they will approve it. So it's a huge amount of data is being submitted to regulatory authority, which is FDA. Okay, phase 4 trial is not a trial. It is just a marketing and surveillance, whether in larger when it goes, because we tested the compound on 3,000 people. Now you are going to give to billions or millions of people, whether it's still there. It does not have any problem. So this is just a monitoring. We call it pharmacovigilance also, in other term. Identify how well treatment works. Monitor long-term benefit and risk. Look for the other uses of drug. And I talked about repurposing this morning. Unsuccessful phase 2 can result in withdrawal of drug from the market. And I will give an example in one of my lecture of why it was withdrawn and why it happened. So this is also an important part of the drug discovery process. So this is my final slide. So what we have to do means to get there. This is a Paul Jensen, who was the chairman of Johnson & Johnson, the parent company of Johnson & Johnson, and he was organic chemist. And I just want to tell you one thing, that when the day he died, he was running a reaction. His reaction was running in the hood, and he was a multi-billion-dollar company owner, actually, but he was still working in the lab. And I have seen a couple of people in Pfizer when I was postdoc there, and they set up the reaction. These are my executive director. They set up the reaction, and sometimes they forget, and they call me, and they say, can you work it up? I forgot. And I said, what it is? And I said, oh, this was this, if you can get something out of it. So people are there. They are not just sitting in their glass offices and they are doing. This person was one of them. That the day he died, he was running a reaction in the end, he died by heart attack. So he says, I always mention this quotation, that a good scientist is someone who succeeds in getting the different scientific discipline to work in harmony with another. And what we talked today with Honorable Vice Chancellor today, this is the same thing, that we have to come together, all discipline in AMU, from engineering college, from chemistry, from INC, come together and just make something happen. Thank you so much for listening. I think I did not over. Only exactly 40 minutes. Okay. Okay.
So you can have a, yeah.
I can answer from there.
Okay, yeah.
Question. Okay.
Yes, sir. Thank you, Dr. Islam, for the informative and well-explained session. Moving ahead, we now invite Dr. Muhammad Azhar Aziz to deliver Lecture 2.
I'm here. Thank you, Zainab. I think let's do a little bit of warm-up. Why don't you guys all come in the front, sitting in the back? This will also move your muscles and post-lunch high glycemia. Come on, just rise up and come in the front. Where do I put this? Here. Nobody wants to move, huh? Lunch was heavy? Ah, yeah, it looks like salads only, huh? Okay. All right, so this is going to be the second lecture for today, and I would love to have an engaging class. See, we are doing a coursework, so either we can decide it to be a monologue, like I speak, you listen, or it can be an interactive session. So wherever you want me to stop, you can stop me, ask a question, and let me tell you one thing. No question is a simple or a stupid question. No, every question is a good question. So feel free, because you all are from different backgrounds. आप सबके disciplines अलग हैं. हो सकता है कोई चीज़ जो आपको बहुत simple लग रही हो, वो उसके लिए बहुत advanced हो. तो इसलिए feel free to stop me anywhere, ask a question, and if you don't understand, you can continue, right? Another thing, you'll see some of the slides repeating between my lecture, Dr. Zainuddin's lecture, and Dr. Madhu Islam's lecture. So this is on purpose. Consider it as a revision. Plus, you'll also find something new, new flavor of the same thing. Like he talked about clinical trials, I'll also tell you about the clinical trials. But you will see something additional to it. Plus, it's a revision, so you will remember it better. Right? Can we start now? Nobody moved? So I remember when I was in Case Western, so they used to get up and do some exercise and then again sit down so that you're having full attention. Otherwise, the attention span these days is very low. So keep it engaging, huh? So I'll be talking mostly from the multidisciplinarity point of view that I started in the morning as well, that it's not just about one man's story. It's a team's story. A team is going to do it. And here's an outline of my content so that you don't get lost. This is how I will navigate. A little bit of introduction, some approaches, methods, phases of drug discovery, drug delivery, some of the introductory slides on that. What are the novel drug delivery systems? Some of the latest breakthrough, AI. Everybody is trying to link to AI. Somehow, AI is going to pervade into every aspects of our life. So we'll touch a bit on that as well. And we'll have some conclusion as well. Okay. So we'll start from very basic. What is a drug? Anybody? It's written on the slide, but anybody can answer that. Yes? Okay, good. Anybody else? What is a drug? It could be for prevention as well. So any substance other than food that is used to prevent, diagnose, treat, or relieve symptoms of a disease or abnormal condition. So that's called a drug. And as Dr. Imad said, drug is not discovered, but I'll try to put it in some of the slides that yes, sometimes you're fishing around and you do find the molecules which can qualify as a drug. So drug discovery is the process through which potential new therapeutic entities are identified. So new molecules, new entities, new biologicals, new materials are found from different sources. They can be synthesized in the lab, or you can even get it from a plant. You know about metformin, a very popular drug for diabetes? It's of plant origin. A lot of drugs have got their origin in the plants. Aspirin, cinchona bark. There are so many examples that have got their origin in the plants. And many of them are serendipitous. They were chance discoveries. So they stumbled upon this molecule, they found its potential, and then they qualify as drugs. And the second aspect is the delivery. You found something, it's very nice, very important, very well working, but it cannot go to in the body, it cannot be tolerated by the body, it cannot be delivered to the target organ. So even if you have something, it cannot be delivered, it's a waste. So delivery is another aspect that is as important as drug discovery. So it's a yeah, nutraceuticals are definitely, but they don't qualify as drugs. That's why they don't want approval. They don't need any approvals as well.
So you don't need approval for them.
Yeah, nutraceuticals
But
over the counter.
they are also acting as a drug, food as a drug.
Yeah, but as we know that it doesn't go through the same rigor of drug testing and
that's why it is easier to sell them, sell those things in the market.
Yes, those are nutraceuticals. That could be like a gray area between drugs and foods.
Gray area between drug and this.
So drug delivery is a method or process of administering a pharmaceutical compound to achieve a therapeutic effect in humans or animals. Now, why multidisciplinary approach is required? Because it has got different aspects. It involves physicians, it involves biologists, it involves understanding of the human physiology, human body, and a very diverse discipline, which is chemistry. They are building molecules in the labs. They know about the synthesis, they know about the pathways of synthesis, but they have got no idea about central nervous system. They've got no idea about the cardiovascular system. So they have to come together. There are so many disciplines that are now getting integrated into a drug development process. So modern drug delivery and discovery is too complex. So it cannot be done by a single person. And you will see in all my slides, this is a repetitive message. Just to break that mindset, which I said in the morning as well, that I am a particular expertise and I can't do that. No, you can. All you have to do is to go and shake hands. That's it. You can do it. So there's a need for genomics, transcriptomics, proteomics, chemistry, AI, pharmacology, and that is how we can increase our rate of success of discovering or finding new drugs. Now, drug discovery timeline, as Dr. Imadul Islam has already mentioned, but do you see a difference? Do you see something new on this slide? Anybody? Anybody tells me the answer gets an extra coffee tomorrow in the morning. कोई तो पता है भाई कोई नई चीज़ है इसमें जो अभी आपने सुनी? Phase 0, exactly. So phase 1, 2, 3, 4, very popular. Now there's a new terminology that's coming up, is phase 0. And you have already seen these slides, like what is discovery, and then preclinical, which is in the animal models. Then comes the phase 0, which is called preliminary trial. The difference between phase 0 and phase 1 is given here. There are 5 basic differences. It's about the proof of concept is there only in phase 0, whereas there's early data that is available for phase 1. Number of participants are different. And then you also have the method of production of these molecules. It could be GLP, GMP method. GLP is used necessary for phase 0, whereas GMP is mandatory for phase 1. And then you also have got the time span, which is about 10 months in case of phase 0, whereas in case of phase 1, it could be 2 to 3 years. And the results are, of course, different. So it may not be visible to you here, so don't worry, don't bother about writing it all. All these slides from the entire course will be given to you, right? So don't worry about if it's not visible here. So we have got these stages, phase 0, 1A, 1B, where we are testing the safety and maximum tolerable dose. Then we go for efficacy, and then phase 3, phase 4, which Dr. Imad has already told in detail, so I'm not going to repeat that. And the terms that are here, maximum tolerated dose, single ascending dose, this is another model where you keep on increasing the dose and see where is the maximum limit a person can tolerate the dose. And there's a variation of it that is called multiple ascending dose, where you have got multiple subjects. It's not in the same subject. It's different subjects with different ascending doses. So you collect data from different subjects rather than a single person. Now, how do we do this? These are the trials that are coming together, but there are 4 different methods that I can identify and I can classify that how this drug discovery is being done. Random screening is like fishing. You have got a library of molecules, and you are screening for activity, you are screening for targeted effect, and you might be lucky, you might find something. I think the Venn diagrams that you showed, that was excellent. Said that there has to be a molecule which qualifies in all 3 criteria. Then only it qualifies, and it's something like a fishing expedition. You might be lucky. Now it has been facilitated by AI. So earlier there used to be assays, people are running 384 well assays and finding results, and then it was literally a fishing expedition. Now you have got fishing navigation. Has anybody seen the navigation tool for fishes? अब iPhone पे एक ऐप है, उसमें आपको बताता है कि मछलियाँ कहाँ ज़्यादा हैं. So you can go and hook there, and आपका fish catching जो है वो आसान हो जाती है. So same thing is even in the drug discovery. Now with the computational tools, we can narrow down our area of search. So that's with random screening. Then we know a molecule that is already there. We can do modifications. Like we have published a couple of papers where there were iso-indigo dyes. So we did a bromine modification. We added a bromine atom, and we also added a chlorine atom. And we did a modification, and we saw different effects on cell death. So that's called molecular manipulation. Then there's molecular designing, where you're starting from the scratch. You design the molecule, and then and this is also under the rational drug design. So you have got a hypothesis, you know the effects of the chemical entities. So you design the drug up front, and then go and see the efficacy of how it behaves. And then it could be drug metabolized, like there was a drug, but then there's a pathway through which it breaks down. So the intermediates can also work as drugs. So after the drug is metabolized, secondary stuff is formed, और वो भी drug की तरह काम कर सकते हैं. So these could be the 4 methods of drug discovery, right? And so this is a continuous procedure starting from target selection. So you have to identify first, what am I going to target? किस चीज़ को target करना? It could be a molecular target, it could be a cellular target, it could be a genomic target, it could be a transcriptome. Are you familiar with these terms? Genomics क्या होता है? Transcriptomics क्या होता है? Anybody who knows transcriptomics? Genomics? Either the lunch was too low or too high, I don't know. Genomics or transcriptomics? Okay, so genomics is all about DNA, right? And the DNA transcribes into RNA, which is transcriptomics. And then RNA translates into protein, that is proteomics. So you'll see it in my subsequent slides. So once you have the target in mind, then you go for the lead discovery, which Dr. Imad also said about, that you try to find a lead molecule. Lead molecule is among a galaxy of molecules. You identify the molecule which is going to be the most active and most target specific. And it can involve all these synthesis and isolation, combinatorial chemistry, then you have to have an assay to see its activity, whether it's active or not. And we also use a high throughput screening. Medicinal chemistry come into picture where in their lab, they're producing molecules which are having activity, which don't have activity. This is tested later on. And these libraries are available for anybody to buy or get it and use it for their purpose. So there are millions of molecules that are available as libraries which can be tested for their potential as drugs in a particular disease. Then we also have structure activity relationship studies, in silico screening, chemical synthesis. This all comes under medicinal chemistry. Once you have the compound, we go for in vitro studies. That is what we normally do in our labs. I also have a lab where we do the cell culture studies. It's mostly grown in 2-dimension, or you can also go for 3-dimension organoids, spheroids, etc. Or you can take it to the living models. These are in vivo models, preclinical models, models that are breathing, models that are having life in it. So what comes to your mind when you talk about in vivo models? जब भी ड्रग टेस्टिंग की बात होती है, चूहे सामने आते हैं. उन्हीं पे सबसे पहला जुल्म उन्हीं पे होता है. ठीक है, छोटे चूहे, बड़े चूहे. Then we go into even non-human primates. अब खैर वो चीज़ें बहुत बदल गई. So animal models are also very much restricted. Now even FDA is talking about removing all these animal models and having some computational models. कल आप लोगों का tutorial होगा, उसमें आप जाके देखेंगे. They have replaced the actual animals with the computational based models for testing all this. So I hope you will enjoy that pharmacology models. From there we go into clinical trials. Yes, please. True. Yes, so in computational models, they are even recreating the physiology. I know a group, and I was part of that network in Harvard Medical School, Center for Virtual Tumor. There was no tumor. They developed the tumor as it grows inside the body of a human being. It was having hypoxia, जो center में उसके low oxygen content होता है, उसमें वो भी था. Flow of the nutrients. It was also having its growth kinetics, and you can see that physiology on your computer. But that was an effort of a full Center for Virtual Tumor. So you can check it out online. So I know you're right that it cannot replicate the physiology of a human being or an animal, but yes, they try to filter it and they try to come as close as possible. So it will not replace, but yes, it will help, it will complement. But who knows? I'm a big proponent of technology. Self-driving cars, it was just science fiction, right? ऐसे कैसे हो सकता है? Self-driving cars कौन बनाएगा? ऐसा हो ही नहीं सकता. But it's a reality now. So coming to the first phase of the slide that we saw, the target selection. This is one of the very important aspects to know what you want to develop the drug against. किस चीज़ को target करना है आपको? In fact, the next course that is coming up, it's all about target identification. कि target तो मिलेगी किसके खिलाफ हमें drug बनानी. अगर वो target मिल जाएगा, तब हमारा अगर सही target हुआ, target से मुराद ये है कि कौन सी ऐसी चीज़ है जिसकी वजह से हम जो disease है उसको reverse कर सकते हैं. जैसे अभी आप लोगों ने सुना होगा कि कुछ scientists ने switch निकाला जिससे कि tumor cells को वापस normal बनाया जा सकता है. So they found a target by which they can revert the tumor into normal cells. So they're not killing the tumor cells, जो कि general approach होती है कि भाई tumor cell इसको kill किया जाए. No, they found a target that can reverse the tumor cell into a normal cell. So identification of target is very, very important. If your target is not correct, I mean, the entire story is going to fizz out. So that's why we can see that most of the targets here, the pie chart that you're seeing here, which has got the highest proportion? Enzymes. So because they are the active proteins, they are the molecules that are affecting the activity of the reactions. So they are delivering there. And the second next is GPCRs, G protein coupled receptors. These are located on the membrane of the cell, and they are easily targetable. So we all know about the DNA, RNA, protein central dogma, right? तो एक वक्त ऐसा था कि we were proponently believing कि अगर सारी चीज़ों की जो information है वो DNA में है, अगर DNA में कोई खराबी है तो वही चीज़ बनेगी disease. तो everything was in DNA. People zeroed down कि हमें अब solution मिल गया. DNA सही कर दिया, चीज़ सही हो गई. But DNA को target करना is not easy because it's located inside the nucleus. कितने सारे barriers cross करके आने होते हैं. पहले क्या क्रॉस करना है? सेल मेम्ब्रेन, फिर इसके बाद न्यूक्लियर मेम्ब्रेन, पहले पे साइटोसॉल से पास होना है, और फिर इसके बाद न्यूक्लियर मेम्ब्रेन, एंड देन यू गेट इनटू द डीएनए, और डीएनए भी टाइटली पैक्ड, that is also not accessible everywhere. So that's why the target that is on the surface, which are GPCRs, G protein coupled receptors, they are 7 helix membrane proteins, and they are easy targets. And then you also have ion channels, which are also available on the surface, and then other receptors are there. Receptors are all very easily accessible. So जो दरवाजे पे खड़ा हुआ है, उसको गिराना आसान है. जो घर के अंदर है, उसको और मुश्किल है. जो घर के अंदर एक लॉकअप में अपने आप को बंद किया, वो high security place. You cannot touch that. So in the target selection, we have got cellular and genetic targets, and then we take help from genomics, proteomics, and a lot of data analysis which comes in the form of bioinformatics. So in case of cellular and genetic targets, this is a lot of basic biology that goes into understanding the disease mechanisms, कि disease काम कैसे कर रही है. जब उसका mechanism समझ में आता है, तो फिर उसके targets मिलते हैं, cellular genetic targets मिलते हैं, right? इसमें receptors, enzymes, जैसे we already talked about that. So we have got the key biological functions पता लगते हैं, and then it could be the disease organism. जैसे अगर हम antibiotics की बात करें, तो we are trying to find out कि बैक्टीरिया के जिंदा रहने के लिए कौन सा जीन important है, कौन सा mechanism important है. So we disrupt that, and then we are able to get rid of that bacteria, right? So we have to understand those basic mechanisms and biology, which can give us the idea about the cellular and genetic targets. But we also are coming up with a lot of efforts, and the data generation has become quite easier in case of genomics and transcriptomics. So now entire genome can be sequenced in about $100. In China, you send out your human genome, it could be $100, $200, $500, you can entire human genome can be generated. So that is creating a lot of enthusiasm in finding out the targets using the genomics or the transcriptomic approaches. And the advent of this technology, CRISPR. So if there is a gene that is solely responsible for causing the disease, we can fix that portion of the DNA and make it healthy. Sickle cell anemia, I think we know about that story. Sickle cell anemia was cured by fixing a single base using the CRISPR-Cas9 technology, which is a gene editing technology. And this is what something that came out of my lab when I was in KMAR, that we identified some gene targets by using the genomics approach from the cancer patients. So there were 11 genes that we reported that were novel to be involved in colorectal cancer. So it's coming out of the basic biology. So genomics as well as transcriptomics, both are nucleic acids, and they complement each other. But the actual function is being done by the proteins. So the protein guys, they say that no, whatever is in the DNA or RNA it has to become a protein to be functional, because these are the functional entities. So if you handle the protein, if you tackle the protein, then we have found the right target. So proteomics, therefore, is also gaining traction. Unfortunately, the level of data generation as it is in genomics or transcriptomics, we haven't reached that in case of proteomics, or there's another omics called metabolomics. We are not yet there, right? But all this data is coming out and it's deluging. It's non-informative unless we use these bioinformatics tools. We need to have the computational power to make sense out of this data. 3 billion bases, right? How are these 3 billion bases are arranged? You will know. You send out your genome, you will find out how are they arranged, what is the sequence. But what does it mean? It's like you are given a book in a different language and you are asked, what is it? So you need to have some kind of synthesis method that you can synthesize the information and bring out a target out of that. And that is being done by the bioinformatic tools. And that's why this is a very upcoming field which involves the data analysis, interpretation. It also involves simulation. So like earlier, you have to do the assays. कि drug आपने बनाई और एक target है आपका, दोनों को आपने attach करा के देखा कि activity आ रही है या नहीं आ रही है, उसका कोई output है या नहीं है. But now you can do this computationally. You can do the simulation experiments, and you could do the molecular modeling. You can do the docking stuff. And you can also do the pathway analysis that, okay, if we disrupt this particular step of the pathway, what will be the effect on the end product? And even the clinical trials, especially there's one type of clinical trial I missed here, NF1 clinical trials, where only one patient is there for the trial. And that needs a lot of bioinformatics support. And this is what we are pitching as personalized medicine, that nobody is the same as the other person, everybody is different, and therefore we need to have a personalized medicine approach. This is possible only when we have the power to do a lot of data analysis. Again, coming back to the self-driving cars. Self-driving cars, you know, what is the main component? Sensors. Sensor उसके ऊपर लगा वो 360 डिग्री डेटा कलेक्ट कर रहा है. सिर्फ डेटा कलेक्ट नहीं कर रहा, उसको रियल टाइम में प्रोसेस करके और फिर इसका इन्फॉर्मेशन भी दे रहा है, and then it's guiding the car for navigation. So it's tackling the situation as well. Cyclist आ गया सामने तो क्या करना है, pedestrian सामने आ गया तो क्या करना है. So this is all possible because now we have the tools and means to do computation. पहले this was not even possible. I'm not sure if you are from the generation of floppies, but I'm sure you are the generation of GBs, right? You have not heard about KBs and MBs. You have heard only about GBs or TBs. So that's why this bioinformatics-based solutions are possible only when we have gone a lot into our computational power increase. So far, anybody can tell me what is target identification? Yes. You can sit down and tell us. Target identification. What do you mean by target? Okay. Anybody else? What is the target? Does it necessarily have to be a protein? Can't it be it could be DNA as well. Okay, so target is any molecule that, if disrupted, would help in preventing or curing a disease. That could be a simple definition, right? Then we go to the lead discovery. So these are the molecules that are going to be said as the lead molecules, and we have got methods for doing the discovery from plants, or it could be chemical synthesis, as we just talked about, or it could be done computationally. It could be coming from the endogenous ligands, which are having some kind of receptors for disruption in the activity or enhancing the activity. So it's not always about disruption. It can also be about, because like in cancer, you've got oncogenes and tumor suppressor genes, right? So for curing the cancer, you can either enhance the expression of the tumor suppressor or bring down the expression of the oncogene. Both ways. Either you take care of the brake or you take care of the accelerator. Ozempic, we are talking about Ozempic, Mounjaro. Ozempic is an GLP-1 agonist. Agonist means it's enhancing its function. Antagonist is it's inhibiting the function. So it could be on both sides. And this needs to be refined by optimizing these chemical hits for clinical trials, which is known as lead optimization. And this is done through doing some potency, selectivity studies. ADME is absorption, distribution. M is for metabolism. E is for excretion. So this defines, these are the 4 parameters on which we see the efficacy of the drugs, and it also is a qualifying criteria for the drugs to move to the next stage. And oral availability and pharmacokinetic properties, this is an additional thing. Like if a drug is orally available, the compliance is more. Like my boss in US, he was able to develop a oral format for 5-fluorouracil, capecitabine. So when he developed this oral formulation, it was better tolerated and compliance was also better. So, and you know that in vaccines, particularly this oral availability is a plus if it is given to the small infants or kids. I think somebody asked the question about the plant-based vaccines. That's how the plant-based vaccine came into picture. That the promise was that plant-based vaccine will have more compliance and they will be having oral route. And for the lead discovery, we have got synthesis and isolation step, combinatorial chemistry, and then you have got to have an assay. Assay development is as important. If you can't measure, how would you know? How would you improve? So for that, we definitely need to have an assay. Assay means some kind of a protocol which can tell us about this particular molecule's activity. It could be a color, we'll be talking about this, it could be in the base of color, absorbance, or it could be some kind of a chemical reaction that can allow us to measure the activity of that molecule. And it is achieved through then we go on to the high throughput screening because we have to screen a number of molecules. So it has to be scalable for high throughput screening, which is known as HTS. Any questions so far? No questions? Okay, so for synthesis and isolation, could be through the separation of mixture. We have got, if you extract something from the plant, is it a single molecule? No, it's a lot of molecules. So you can start with the separation of mixture, then you remove the impurities, or it could be in vitro chemical synthesis. As I said, from the scratch, you can design the molecules, or you can just play around with the atoms for chemical synthesis, or it could be the biosynthetic intermediate, like metabolizing the drug and finding those intermediates. The examples of this is polyketides and alkaloids. Assay development, we just talked about, is used for measuring the activity of a drug. Without an assay, even if a drug is wonderful, it's going to the dustbin. So you have to have an assay so that you can compare, you can prove its efficacy, and you can prove its activity. And for that, we need to evaluate the expressed protein targets and how is the enzyme substrate interactions are going on. And it also is the kind of filter, whether this molecule should go forward or it should be killed right here. So if an assay cannot be developed for its activity, it could be killed right there. It has to be scalable. For that, it has to have a high throughput screening capability, and so that we can run 3-4 well plates or 96 well plates. And now this is necessary to have robotic-based setup where the human intervention and human errors are minimized, rather it's being taken care of mostly the robotic arms. So then we have got the different assay technologies in high throughput screening. We can have the cell growth based assays that we commonly do in our labs. We grow the cells and see आपने petri plate पे cells को grow किया. You put the compound and see whether the cells are growing more, less, getting killed, or they are alive. The MTD assay. How many of you are familiar with MTD assay? किसी ने MTD assay किया है? Nobody? Okay. Then tissue response. This is functional cell-based assay and also the enzyme test. Enzyme test किए होंगे? Lipase वगैरह chemistry labs में. And the methods that we commonly use for these HTS, it could be spectroscopy based, it could be mass spec like we talked about LCMS and MS and GCMS, gas chromatography or liquid chromatography, or it could be calorimetry based, it could be XRD, X-ray diffraction, or simple imaging based using the microscopy or the radioactive methods as well. How much time do we have? So in the medicinal chemistry aspect, we have got, this is the discipline which is in the intersection of synthetic organic chemistry and pharmacology. And it goes for, and Dr. Imadul Islam has done a lot of medicinal chemistry. He is known as a medicinal chemist. So, and I think your work was on antibody drug conjugates, right? So you have done a lot on those molecules and discovered new molecules or formed new molecules that are in the clinical trials as well. So this is one of the very cornerstones for the entire drug discovery industry that involves drug discovery, lead optimization, process chemistry and development, and this is hypothesis-driven optimization. So if you have got a hypothesis, you have got a target, then it becomes easier than the fishing expedition. So can you see the difference between fishing expedition and target-driven, hypothesis-driven selection? Do you see the difference? एक तरफ आपने मॉलिक्यूल्स बना के रख दिए, अब आप उसको टेस्ट कर रहे हैं कि शायद ये इसमें से कोई एक्टिव हो, शायद इसमें से कोई किसी चीज़ में काम आ जाए, और दूसरा ये कि आपको पता है कि ये मैकेनिज्म है, ये रिसेप्टर है, इसको टारगेट करना है, इसके लिए हमें मॉलिक्यूल डेवलप करना है, इसके लिए एक्टिविटी चाहिए, ये मॉलिक्यूल चाहिए. So that's the difference. So it involves library development, structure activity relationships, in silico screening, and chemical synthesis. Library development, you can have a lot of chemicals and you play around and get those molecules ready. And whenever you have to start, you can pick up those molecules either through rational designing, you can filter it out initially and then go over for screening. Or if you have the capacity and resources and money, you can have a large number of libraries screened. Then you also have chemical synthesis, where you can do the synthesis of your own molecules and do some structure activity relationship. And this is where the in silico screening comes into play, and that helps. That you do the computer screening of the molecules, and you find out the structures, you see the binding of the target with the drug on the computer, and see the effect on the target as well. And that filters a lot of enormous chemical space. So I think now this is a standard that you do these kind of studies before you go into doing the actual assays, and you do that involves the reagents, the manpower that are going to be a waste. So here I have listed some of the softwares that I use to perform docking and visualization. And so these are some of the softwares that you would find useful if you want to try your hands on. There are more. I have just listed here 6 of them. And we recently had a paper from one of my MTech students where he used this ChemDoc in Vienna, and he found the PD-L1 inhibitors. So he took the PD-L1. Anybody familiar with PD-L1? Anybody from the back? Just speak so that I know nobody's sleeping. PD-L1 is a receptor that is helpful in immunotherapy of cancer, which is now one of the most successful strategies. So computational identification of PD-L1 inhibitors was done by using these gold and silver nanoparticles, and we were able to conjugate the drugs and see in silico, with the help of in silico studies, that what will be the effect on this particular target. With the in vitro studies, so now once we do the computational studies on the screen, we wear the gloves, we wear the lab coat, come in the lab, and we do the in vitro studies, where we have got the things on the Petri plate or a test tube, right? And this could be the cell lines. How many of you have seen the cell lines? Which cell line? Cell lines are the cells that are derived from the human body or the animal's body, and then they are propagated in the lab. So tomorrow, in the tutorial, you'll also see this snapshot of what are cells, how are they propagated, how are they grown outside the real environment. Or it could be some subcellular components like ribosomes or mitochondria. If your target is in mitochondria, you will definitely need to culture that. Or it could be the extracts, like in case of plants, it could be a wheat germ extract, or it could be reticulocyte extracts. So extracts are giving you the entire cellular contents. So advantages of in vitro experiments are that, first, the biggest advantage I see is you don't have a lot of regulatory approvals needed. You can run as many experiments with the cell lines as much you have. अगर आपको rats पे भी करना होगा, rabbit पे भी करना होगा, या mouse पे करना होगा, you need to have ethical approval and all that. Whereas in case of in vitro, you don't need any of those approvals. That's the biggest advantage I see. But it's also cost effective. It's faster, like MTD assay is done, you get a result in 24 hours that you have got all the results. But the disadvantages, what could be the possible disadvantages? So it's in vitro. The disadvantage is it's not mimicking the actual scenario. We are trying to mimic the actual human body. We are going to put something inside the human body. So we need to have an environment which is as close as possible. So we gradually go there, and this is far from there. This is only the first step. So how is it done? There are some static models where you have got the 2D cultures, 3D cultures, or spheroids, organoids. These are all called static models. Then you have got the static microphysiological systems. What you're seeing here is now in the microfluidic stages. These are the chips that are available in which you can do the co-cultures. You can have multiple cells. क्योंकि एक तरह के सेल एक जगह पे नहीं ग्रो करते हैं, बल्कि काफ़ी सारे सेल्स एक साथ ग्रो करते हैं. So you can do the co-culturing experiments. And you can also have a dynamic MPS models, which is now set as organ on a chip. So you can have mimicking an organ. Organ has got multiple entities. कई तरह के cells होते हैं, कई तरह की उसमें blood supply होती है. वो सब you can put it on a chip on a micro scale, and that can be used as a model for testing your drug. So this is the one that can be used for assays. जो हमारा lead molecule है, उसके लिए assay development. Then you also have the in vivo studies. Now we are from the plates and tubes, we are moving to the actual breathing animals. But still, they are not humans. बहुत सारी drugs शुरू होती हैं, बहुत अच्छा लगता है स्क्रीन पे computationally. जब हम उसको टेस्ट करते हैं पेट्री प्लेट पे सेल्स के साथ, फेल हो जाती हैं, कोई एक्टिविटी नहीं आती है. फिर उसके बाद वहाँ से जो मॉलिक्यूल्स निकलते हैं उनको हम एनिमल्स में टेस्ट करते हैं, वहाँ फेल हो जाते हैं. तो इट्स काइंड ऑफ फिल्टरिंग शुरू हो जाती है. और बहुत सारे मॉलिक्यूल्स जो बहुत अच्छी एक्टिविटी दिखाएंगे एनिमल्स में. बहुत आपने स्टडी सुनी होंगी, ट्यूमर ग्रो किया माउस में, इंजेक्शन डाला, फौरन श्रिंक कर गया, वाह, कैंसर की दवा मिल गई. लेकिन जब वो आता है ह्यूमन ट्रायल्स में, फेल्स. And that happens a lot. That's why the attrition rate for drugs in the pharma industry is very, very high. I hope you'll agree with that. That attrition rate is main cost that we are paying for high, these expensive medicines. Billions of dollars लगते हैं ड्रग को बनाने में, उसमें attrition rate यानी failures बहुत ज़्यादा है, उसकी भी cost जो consumer उसको ही pay करनी पड़ती है. So in vivo studies, we are using the whole organism. It gives us the information about metabolic profile, toxicology, drug interactions. But as I said, there are ethical concerns, cost and time. Each animal is costly, and it also involves a lot of time. It takes months to generate data. Then we have got the different models, animal models for different disease states. You also have the genetic models. For example, in cancer, there's a gene P53 that is mutated, right? So if you are working on something that is having a P53 wild type gene, it's not going to have an actual scenario. So you need to have those mouse models which have got P53 mutation. So this is another branch of another discipline that is working on preparing mouse models, animal models that can ensure that the drug will be successful in the human beings. So I think by now, how many disciplines have you seen that are involved in drug discipline? Can somebody do their memory test? Yes. Good. Bioinformatics, biology. फिजिशियन अभी नहीं आए पिक्चर में, वो आएंगे जब हम क्लिनिकल ट्रायल शुरू करेंगे, वो अभी तक नहीं आए, आगे आने वाले हैं. यस जी, मेडिसिन. ओके, यू मीन द फिजिशियन फ्रॉम द मेडिसिन, मेडिसिनल केमिस्ट. ओके, यस. हु एल्स? माइक्रोबायोलॉजिस्ट. यस. प्लांट्स वाले अंडर रिप्रेजेंटेड हैं यहाँ पे. प्लांट बायोलॉजिस्ट, प्लांट मॉलिक्यूलर बायोलॉजिस्ट, प्लांट फिजियोलॉजिस्ट, जिनको पता है कि अगर पत्ती को आप रब करते हैं तो कुछ एसेंशियल ऑयल्स निकलते हैं उसमें से, और उन एसेंशियल ऑयल से दवा बन सकती है. दिस इज समथिंग दैट ओनली अ प्लांट बायोलॉजिस्ट कैन डू. सो as we move forward, you will appreciate that there is a lot of disciplines that are coming together for making a drug. They are involved in this. So these are the in vivo studies where we do even the behavioral studies, functional imaging, and we also do the ex vivo studies. I'll come to that. And these are different models. As I said, it could be as simple as a Drosophila. Drosophila is a fly which is very close to have the human genetic setup. And I'll tell you a story about my collaboration. There was a scientist in Mount Sinai, New York. They had developed these Drosophila-based fly avatars. And he made a library of them. So, and you can use them for testing your lead compound. And it was so good that it was something similar to doing a clinical trial. And now it has been taken up by a company in London. They have acquired this technology and they are marketing even in Saudi Arabia because it's highly expensive. And we have got other models like zebrafish and mouse model you all are familiar with. So we have got in silico, in vitro, in vivo, right? So we have gone through all these three things that are used for the assay development. Then there's ex vivo. Ex vivo means outside the human body. So have you heard about CAR-T cell therapy? So the information is taken out from the patient itself, and then those cells are engineered and reprogrammed. For example, their immunity is increased and then put back into the patients to fight. So especially in the case of cancer, and this is highly personalized. Every patient is different. So the patient cells are taken out, engineered, reprogrammed, put back into the patients. Now they are able to fight the cancer and get cured. And this is one of the most transformative therapy in cancer. This is the most promising therapy in cancer. So these are ex vivo models. So why ex vivo studies are used? Because they are the gap between in vitro and in vivo. So they give you the best of the both worlds. You can do it outside the body in controlled conditions, and you don't have to do it inside the body. Like Robert Jarvik. Have you heard about Robert Jarvik? The inventor of the artificial heart. So एक बार वो अपनी कार ठीक कराने के लिए गया मैकेनिक के पास. मैकेनिक पूछा आप क्या करते हैं? कहने में हार्ट स्पेशल. हार्ट क्या होता है? एक हार्ट ऐसी जैसे तुम्हारी कार का कार्बोरेटर होता है ना, वैसा होता है हार्ट. तो कहने तो कौन सा बड़ा काम करते हैं आप? हम तो कार्बोरेटर रोज चेंज करते हैं. तो उन्होंने कहा ठीक है. कार को स्टार्ट किया, ऑन किया, कहने अब चेंज करो इसको. अब इसका कार्बोरेटर चेंज करो. कहेंगे ऐसे कैसे चेंज कर सकते? बंद तो कीजिए इसको, तब तो हम काम शुरू करें. कहेंगे हम बंद नहीं कर सकते. हमें उसी जो है जीते जागते इंसान में थेरेपी करनी है. हमें उसी में हार्ट ट्रांसप्लांट करना होता है. तो दिस इज अबाउट अ लिविंग ह्यूमन बीइंग. उसमें ही हमें थेरेपी करनी है. सो वी आर ट्रीटिंग अ ह्यूमन बीइंग बाय टेकिंग इट्स ओन मटेरियल, engineering it outside, and then putting it back. Right? And it's good for personalized medicine. Then we have got, so far we have covered introduction, drug discovery approaches and methods, drug discovery phases. Any questions so far? No? Yes, Shahid. He's one of the students who was very good in asking questions. And you know what is his background? Physics. Yes, Shahid. Yeah. Yeah, his question is कि आप एक ह्यूमन बीइंग में ट्रिलियंस ऑफ सेल्स होते हैं. ठीक है, आपने एक सेल का डीएनए ठीक कर दिया, तो बाकी के साथ क्या कैसे होगा? राइट? दिस इज योर क्वेश्चन. एनीबडी कैन आंसर? जी? Okay. Yes, so usually जो इंजीनियरिंग की जा रही है वो उन सेल्स की जा रही है जो कि पूरी बॉडी में सर्कुलेट होंगे. राइट? तो वो जहाँ पे फैक्ट्री है, जो बोन मैरो है, वहाँ पे चेंजेस किए जाते हैं ताकि जो फैक्ट्री से जो नए सेल्स निकलें वो सब तरफ हेल्दी. फैक्ट्री में जैसे चीज़ ख़राब बनेगी वो सब जगह ख़राब पहुँचेगी. फैक्ट्री में सही कर दिया, सब जगह सही पहुँचेगी. Okay, so now we'll just look at the data-driven, because I'm reinforcing the message of multidisciplinarity. Multidisciplinary approach, we have got the candidate molecule in silico, then we have got this pharmacokinetics, safety, efficacy, and all this is now getting a boost by using the AI, by using simulation, by using experiments. Now once the drug is made, the next big challenge is its delivery. अब इसको पहुंचाना है अपने टारगेट तक. आपने बहुत अच्छा सा खाना बनाया, but it's not reaching to the right person in the right format. Zomato पे ऑर्डर करते हैं ना, बहुत अच्छे मेन्यू में चीज़ें मिलती हैं, दिखाई पड़ती हैं, but if it's not reaching the person in the right format, it's spoiled. So the purpose of doing drug delivery studies is to let the drug reach to its desired place in the right format, in the right dose, in the right time, and also getting cleared off. उसको बॉडी में रहना भी नहीं लंबे वक्त तक. So it should be excreted out as well. So that's the whole purpose of drug delivery stuff. So drug delivery is through all the routes that can absorb. It could be oral, buccal, pulmonary, through the lungs, and there are different formulations for different routes of delivery. जैसे कुछ ड्रग्स हैं जो कि इनहेलर के फॉर्म में हैं. कौन से ड्रग्स? जिनको कि लंग्स में जल्दी से आना है. राइट? कुछ ड्रग्स हैं जिनको ब्रेन में पहुंचाना है, कुछ ड्रग्स हैं जिनको स्किन में ही रखना है. तो you don't need to take intravenous. You will need something that is applied on the skin. Then there are nasal, sublingual, depending on the target organ, it could be different. And the one that is going to be everywhere will be the intravenous through the blood, because blood is going to reach everywhere. So these, there are different formulations, there are different materials that are being used for the drug delivery system. Liposome, it's a ball of lipids. Ball of lipids helps to protect as well as go and fuse with the cell membrane, because cell membrane is made up of lipids. So it can go to the cell, fuse and deliver easily. Right? They are same. So liposome, dendrimers are the other stuff, inorganic nanoparticles, lipid nanoparticles, hydrogels, carbon-based nanoparticles, magnetic nanoparticles. These are all packaging. ज़्यादातर इसमें से जो है सब पैकेजिंग है. कैसे पैक करेंगे? जैसे कुछ चीज़ों को पैक किया जाता है कार्डबोर्ड में. कभी आपने ऑर्डर किया हो तो कुछ नाजुक चीज़ें जो हैं वो बहुत अच्छी पैकिंग में आती हैं. बहुत उसमें कई मल्टी लेयर्ड पैकिंग की जाती है. Right? So in this case also there are different materials for different types of drugs. There could be water-soluble drug. There could be the drugs that are water-insoluble. For that we need to have a different strategy. They are not soluble in water. So liposome-based drug delivery, this is what it looks like. It's a ball of lipids, and this has got a core of water. But there are variations of this which are known as solid lipid nanoparticles, which have got the ball, which is where the core is also made up of lipid. So you can pack the lipid-soluble drugs as well. Although it is desirable that we have water-soluble drug, but somehow it's not becoming water-soluble. So we can change the formulation. Packaging बदल देंगे. अगर packaging हमने उसकी कर ली, then we can use it as a drug by using these drug delivery systems. And then we can add some of the addresses, the peptide, for making it specific for a particular receptor, or we can use polyethylene glycol for protecting it from the immune system, or we can add the DNA or antibody for making it more specific. Next is dendrimers. Dendrimers are branched structures. This is a branched structure, and these branches are all having chemical groups that can serve as the attachment point for the drugs. So dendry means tree. So it has got a tree-like structure, और tree की हर branch पे आप drugs को load कर सकते हैं, और load करके then, so they are based on how many circles are there, they are generations. So if you have got only one circle, G0, two circles, it's G1, G2, G3. So far G4 तक लोगों ने dendrimers बनाए हुए हैं. Right? And it has been used for drug delivery in case of eye formulations. So I'll try to zip through quickly. Then we have got the hydrogel-based drug delivery system. Hydrogel is a material which can absorb a lot of water, so it can swell up. So it is in its powder form, it's dry, but as soon as it gets in touch with water, it can swell up, and it can also release on reaching a particular target based on the difference in pH or temperature. So these are the hydrogel-based drug delivery system. I think you'll see, because this is an introductory lecture, you'll see more in detail in the upcoming lectures. So examples are given here, and we can go through that in detail. Inorganic nanoparticles are there, which are used for crossing the blood-brain barrier. Blood-brain barrier, it is a natural mechanism to prevent the unauthorized passage of molecules into the brain. It's a very tight junction where not everything can pass into the brain. So for passing through that barrier, there are certain inorganic nanoparticles that can be utilized, and they can go through that. And it's also for the ocular drug delivery for the eyes, cancer theranostics. Theranostics are the agent which can be used both for therapy as well as diagnostics. Those are known as theranostic agents. When we have got the carbon-based, so we have got inorganic systems as well as carbon-based. So people have used carbon nanotubes and even the fullerene balls for packing the drugs and delivery as well. So they have all been utilized. They have been utilized in photothermal therapy as well. So I'm not sure how familiar you are with these terms, but I'll be happy to answer, and it may come up in the subsequent lectures as well. Then we have got the mRNA delivery system. I particularly emphasized on this because this is an upcoming field in case of the drugs. mRNA, the COVID-19 vaccine, everybody knows about. It was an mRNA formulation. Some of the companies, they have got the mRNA formulations, and they were protected in these liposomes and delivered. Then we also have the magnetic nanoparticles, which are easy to locate, easy to trace, and also they are being used for the drug release by the application of the magnetic field. And if you come to the recent advancements, I just wanted to mention this guy who's got the Nobel Prize, David Baker. So I'm switching gears into the AI again. David Baker, they recently designed an antibody on the computer. अभी तक एंटीबॉडीज बनाने के लिए एंटीजन को आपको एनिमल में डालना है, फिर इसके बाद इसमें से जो एंटीबॉडीज निकलेंगी उनको फिर स्कैन करना है, देखना है किसकी एक्टिविटी है, कौन सी न्यूट्रलाइजिंग है, कौन सी नहीं है. बट डेविड बेकर की लेटेस्ट रिपोर्ट ये है, आई थिंक 2 weeks ago it was published, that they designed an antibody from scratch on the screen, and it was found to be active. Another upcoming field is click chemistry, which also got the Nobel Prize a couple of years ago, which is a green method of making molecules, and it's very helpful in designing new molecules that could be served for the drug purpose. So we have got drug discovery, drug delivery, and it has to be combined with some bioimaging and material science for taking it to the public. And this is being aided by AI again for data processing as a lab application, and the self-learning models where the data generated and data applied teaches its own model. I think that there's a key applications of AI in drug discovery. In all the stages that you have seen, AI is now helping. I would put that in a simple way, right? And there are a lot of benefits. I think we need to have a fresh morning session to discuss that, because many of you would disagree that AI is not always beneficial. This has got this. So I'll leave it for this would be my second last slide, and this will be the conclusion. So all these slides will be provided to you, so don't worry about that. And if you have questions, we can discuss it tomorrow. So no questions? Yes. Either you have to come forward or speak a little louder. Yeah, see, drug is, vaccine is a type of drug. So it could be using the same mechanisms that we are talking about as drug delivery, like liposome based. It could be the same. They could be oral as well. They could be oral, they could be intravenous, it could be intramuscular, they could be intradermal, subcutaneous. All formulations are there. Ji? Polio is oral, yeah. That's a very popular example. All right, so we'll conclude here. I hope you had a learning and enjoyable session today. Tomorrow we are meeting at 9:30 in the civil engineering department. How many of you are familiar with that place? If not, we can show it to you now. Rest of you are not familiar? सबको पता है civil engineering कहाँ है? अगर नहीं पता तो please अपने को लिए, otherwise we'll post the location also on the group. ठीक है? तो we'll meet there at 9:30. फिर 2 groups बन जाएंगे. एक group जाएगा pharmacology वहाँ पे आप लोग को ट्यूटोरियल दिया जाएगा और एक ग्रुप जाएगा नैनो टेक्नोलॉजी सेंटर, वहाँ पे ट्यूटोरियल होगा, देन विल स्विच। बस अरेंज कर दी है, बट आई लव टू वॉक, अगर आप लोग वॉक करना चाहें तो इट्स अ वेरी ब्यूटीफुल कैंपस, यू कैन गेट टू सी, इट्स ऑप्शनल। वैसे बस अवेलेबल रहेगी, इट विल बी देयर। और कोई अनाउंसमेंट चाहिए? ऑलराइट, एंड सो आई विल सी यू टुमारो देन। आज की सबकी अटेंडेंस लग गई है? Because attendance is mandatory, we have to send the attendance to the Gyan Ministry of Education. कल भी ख्याल रखिएगा अपनी अटेंडेंस का। Used to my next stuff.
I don't know. Ask in the audience. I don't know.
क्यों?
It's attendance sheet. It's a registration sheet too.